having me here. This is a tremendous honor and tremendously intimidating having all these paintings around here. But it's wonderful to be here, and I really thank you for giving me the opportunity to talk to you. So what I want to do today is talk about the work that I've been doing, and as Diana said, I want to highlight the value of islands and what islands have really allowed us to get at, the kinds of questions we can get at using islands. And so I'll paint things in fairly broad strokes, but hopefully I'll be able to convince you as to the value of islands and what kinds of questions we can get at. So first of all, where did it all start? I'm from Scotland, the southwest of Scotland, and what I grew up in the little part down here in the bottom left-hand corner. And I did my degree at the University of Edinburgh. And here I started working on spiders as an undergraduate student working with Philip Ashmole. I started looking at the behavioral ecology of spiders. And so I was interested in behavioral ecology, and I actually wasn't that interested in spiders, if I were truly honest, but Ashmole was just getting into spiders and said, well, here's a project on spiders. Once you get to understand these animals and understand what they have to offer, they're tremendously valuable in terms of providing information on ecology, behavior, as well as evolution. So after I'd finished my undergraduate degree, I came over from Scotland over to the University of Tennessee, and I came there to do my PhD. And the reason was that there was a spider person a person who worked on spiders at the University of Tennessee, and so I went there to study habitat selection in these long-jawed spiders. And these spiders were quite cool because they lived over water, and I wanted to see why they lived over water. I don't know that I ever actually answered that question, but they're very restricted to habitats over water. And they build flimsy webs over water, and they capture midges, mosquitoes, weak-flying insects as they come off the water. So that was kind of, I had no more to do with islands at that point, but there was, after I'd finished my PhD, I heard about a postdoc position that was to look at the behavioral ecology of another spider in Hawaii this time. So this took me right over there to Hawaii. Now, the thing is, I think, maybe I'll try and just play this again, just because the point here is that you get to the coast, and it's a long haul out there. And when I was first going out to do this postdoc, I hadn't realized just the extent of isolation. I couldn't believe it had taken that long to fly over water and reach this little island. And when you think of Hawaii, at least for me, I was not thinking of these spiders, which are the ones I went out there to study. I was thinking much more in terms of the beauty of island systems, you know, the beaches, the palm trees. And to be honest, it's something I never really enjoyed. And so I immediately started to appreciate the isolation. But this was kind of the image I had, you know, walking along beaches, and I thought, well, you know, it's just not my thing. So when you think then about Hawaii, this is the idea of Hawaii, the beaches, the palm trees. And Mark Twain said of Hawaii, it's the loveliest fleet of islands that lies anchored in any ocean. It is truly beautiful, but I'm going to tell you a little bit about parts of Hawaii that are generally not known by visitors. So first of all, I want to just step back a little bit and talk about islands in general. Why do people study islands? You know, Diana said that we're island, we work on islands, we love to work on islands. What's so good about islands? some of the key theories in biology came out of islands, and one that we're probably very familiar with is one by that was really put out in the context of the equilibrium theory of island biogeography by MacArthur and Wilson in 1967. And people had argued about this idea before, but MacArthur and Wilson really formalized it very nicely. But the idea is that the number of species present in any one area, in any one isolated patch, is a balance between immigration, so immigration, things coming in, and extinction, things going out. And so the number of species that's present at any one point is a balance between these two processes. When islands become more isolated, immigration goes down, and the number of species present at equilibrium goes down. When the islands decrease in size, the extinction rate, so the extinction rate will increase, and then, when islands increase in size, extinction rate decreases because they can support more species, and so then the number of species present at equilibrium will then also increase. So area and isolation are key things in dictating how many species you get on an island, based on this balance between immigration and extinction. So that was a tremendously important ecological theory that was based out of islands. However, there are other developments that really relied on islands, and one that we're probably most familiar with is the work of Charles Darwin. And Darwin worked in the Galapagos. Here's old Darwin there. And so Darwin, here's a younger Darwin here. So Darwin worked on the Galapagos Islands and looked at how the finches of the Galapagos had evolved in isolation on those islands. And these ideas were, as you well know, the basis for his development of the theory of natural selection. Now, you come out to Hawaii then. Hawaii, as I said, is extremely isolated. And MacArthur and Wilson talked about this when they were developing the equilibrium theory. And they say near the outer limit of the dispersal range of a given taxon, speciation and exchange of newly formed autochthonous species within an archipelago can outrun immigration. Now, they didn't incorporate this into their model explicitly, but they highlighted how very isolated islands can be the basis, can basically foster diversification. So here you have the Galapagos, the finches diversifying in the Galapagos, and you see these nice little finches that look basically the same, but you don't tell Peter and Rosemary Grant that. So then you come out to Hawaii, and you see here are a similar group of finches, and look what they've done. So they've just diversified in every possible way. So you think, how could this possibly be a finch? And so it's just very extreme radiation, because basically these are the only major group of terrestrial birds that got to Hawaii. So here you can see what people have said is, you know, if Darwin had got to Hawaii, he'd never have left. The thing is, I think that probably if Darwin had got to Hawaii, he'd never have figured out how it all worked. They're just too diverse. And this incredible diversity has really stymied people for a long time. So let's just look a bit more at Hawaii. So as I said, Hawaii is what you often consider this tropical island resort, you know, just a beautiful place. But what I'm going to talk about is the higher elevation habitats where currently you get most of the native and endemic species in all sorts of different habitat types, ranging from dense forests that you see here to bogs and all sorts of different habitats that you get at higher elevation. Just to quote Perkins, and I'll tell you a little bit about Perkins, just to give you a feel for what it's like up in these habitats. Perkins, who collected in the late 1890s, he worked just single-handedly collecting throughout the islands, and says, I was obliged to carry a limited supply of oil in a small oil stove on account of the difficulty or impossibility of making a fire during the continuous and heavy rains for the purpose of cooking rice. This will be as much as the collector will care to pack in so rough a country, and I found the stove and oil a sore burden, very reluctantly assumed. So let's just think about this. This gets at what it's like in these habitats. It's extremely wet, and it's actually very difficult to work. So a little bit about Perkins. Perkins actually was, he came from England, born in 1866, educated in mostly in Oxford, and then he was recruited by the British Association for Advancement of Sciences to conduct a survey of the Hawaiian fauna. So he went out there. He was selected by the Sandwich Islands Committee and set off there in 1891. Now, this man was really remarkable. He spent the next 10 years in the Hawaiian Islands collecting all groups of terrestrial animals. After this, he went on to be hired by the Board of Agriculture so that he could continue on his work in the islands. He was actually hired by the Division of Entomology at the Hawaiian Sugar Planters Association, which allowed him to stay in the islands working on the native entomofauna in particular. He then retired to England in 1912 and was awarded a DSc at Oxford in 1906, the gold medal of the Linnean Society in 1912, elected FRS in 1920 and died in 1955. So why am I highlighting Perkins? Because when I reached Hawaii, he was the man. He was really the only person that had ever collected spiders, at least the most diverse spiders in Hawaii. So just a little bit about him. He spent incredible amounts of time in the islands, and the collections resulted in the Fauna Hawaiiensis in 3 volumes. In the introduction, in which he gives an extensive background into what it's like to be there and what the environment was like, he argued that species arose through a series of events involving divergence and geographical isolation, followed by speciation with competition increasing as new species were added. Now, if you think of when he was working, I mean, this was just, Charles Darwin was just, he'd formulated his ideas, but it was very early in the ideas of evolution. And Perkins actually laid out, purely by observations in these forests, he figured out really what was going on, all of these microevolutionary processes in the islands. He spent many years of field-based research on multiple groups and made extensive ecological observations. So one of the things that he did was he just by sitting and watching, he made observations of the Hawaiian Drosophila, a lot of which have only recently come to light. He said that some of the species are quite conspicuous and are readily attracted by the sap oozing from broken limbs of a tree or from exudations caused by the decay or disease. Very many breed in stems of trees or plants, which when decaying yield abundant moisture, such as those of the arboreal lobelia. So this was hugely important because it's really only recently that we've figured out exactly what the Drosophila are attracted to. They're actually attracted to these exudates, as Perkins pointed out. But just finding which exudates and why is a point of major research at the moment, because they pick up fungi while they're doing this, and there's a whole complex interaction that people are just working on right now. So that was Perkins in a very, I don't think I've given him his full due, but he was a very important figure in early entomology, and I should say in the fauna in general. The next person to come along was this really quite incredible character, Elwood Zimmerman. And Zimmerman had also a pretty remarkable life. He was from California and developed a circle of boyhood friends. Lindsley Gressett, Gordon Lindsley, Robert Usinger, all of whom became eminent entomologists in the early part of this century to the middle part. Zimmerman became intrigued with the Pacific and the insular biota, and so went on as a field entomologist to the Bernice-Papahi Bishop Museum in Honolulu, and became part of the Mangarevan expedition to southeastern Polynesia in 1934. After this, he settled in Honolulu. And why am I pointing all of this out? Because he undertook himself as a single person to write the insects of Hawaii. Now, you think to write on an entire fauna, this is really fairly remarkable. And it's to his credit that he really came remarkably close to doing this. He did a huge amount of work. He was really, in writing this, he was kind of wanted to do what E.O. Essig, who was also at Berkeley, Essig had written the guide to the insects of Western North America. And so Zimmerman wanted to do the same in Hawaii. So he did accomplish this tremendously ambitious feat almost single-handedly, with the help only from Elmo Hardy for the Diptera. And so the Insects of Hawaii came out in multiple volumes. And in 1948, he received a Fulbright to work in the British Museum and spent most of the rest of his life in London, at least while he was working up the Insects of Hawaii. So this is kind of where entomology stood in the kind of the late 50s, early 60s. And then there was a bit of a hiatus until it was really the work of Hampton Carson, an eminent geneticist who I was fortunate enough to know and interact with quite extensively before he died just a few years ago. But Hampton Carson really, he worked with Elmo Hardy, who did the Insects of Hawaii for the Diptera. And Hampton Carson was a geneticist, a dipterist, and understood from Hardy how diverse the Diptera were. And what he also found was that Diptera had giant chromosomes, and you could piece the chromosomes together a bit like a jigsaw, and then figure out relatedness. And this is what this is. It's based on giant salivary chromosomes, and he just pieced together who was related to whom amongst the entire diversity of Hawaiian Drosophila, based on the pattern of the chromosomal inversions. So this was a fairly remarkable effort, and people became really interested in Hawaii, and they said, you know, the Drosophila are just so amazing. And then after that, really because of the Hawaiian Drosophila work, there was this IBP, the International Biological Program, had an emphasis on Hawaii. And this was the effort of Dieter Müller-Dembois in particular, working mostly with Hamp Carson. And they developed papers and research agendas focusing on multiple different taxa, in particular the trees, Metrosideros and Acacia, and then and Myoporum, but also insects, Drosophila, Placothmyces, Scylla, various other insects. And so this was kind of it kind of went plugging along Hawaiian entomology, but still we're relying heavily on the work of Perkins at this point. But it was then in the 70s and 80s that where I was fortunate enough to arrive in Hawaii. There was a renaissance in entomology, really to the credit of three people. Frank Howarth, that you see up here, who worked primarily on cave organisms. This is Wayne Gagne, who died actually not long after I reached Hawaii. And this is Steve Montgomery here. Steve is, it's actually, I'm with him there on the top of Mount Ka'ala on Oahu. And Steve is just pointing out this caterpillar, which the behavior of this caterpillar had been totally unknown, but look what it does. You see, here's this little caterpillar that's meant to just chew on leaves and happily be herbivorous, but what's it doing there? It's eating that insect. And so it was these entomologists that had keen observations and were able to understand the biota in the islands. Now, what about spiders? So this is where it gets very exciting. So I arrived in Hawaii in 1987, and as I said earlier, I came to work on happy face spiders, which were it was one species that was known and had been described. So, first of all, how in the world do spiders get to Hawaii or any isolated place? You think, well, they're fairly, they sit there and they build webs, and how can they possibly get out over thousands of miles of ocean? But this is what they do. They balloon. And so here you see a little spider letting out silk. The wind blows and pulls the silk as it lets it out and blows, and the silk pulls and poof, off it goes. So you see, here it goes, poof, off it goes into oblivion. And so this is how they disperse. So when I got to Hawaii, what I found was a fauna, besides happy face spiders, which I looked at out of the corner of my eye still. But what was really remarkable was that there were all of these long-jawed spiders. I'd done my PhD on long-jawed spiders. I'd done my PhD on spiders that were restricted to water. They lived over water, built flimsy webs over water. And so here I was in the forest, and these spiders were in the forest. And here you get this big fat thing in the middle of the forest. It still pretends to be a stick because it doesn't realize it's a big fat thing. They've got very stereotyped behavior that's characteristic of Tetragnatha. And these are spiders actually from one place, the summit of Mount Ka'ala on Oahu. And actually, all of them are currently still undescribed, which is unfortunately because it takes a long time, as many of you know, it takes a long time to describe these things. This is one that is still undescribed. Just a remarkable animal. It feeds on amphipods. It sits up on the top of the mountain, and it's just got little serrations along the jaws. And what it does is it grabs little slippery amphipods and holds them in these modified chelicerae, the jaws, and just feeds on these amphipods. This is one that had been described. It was described by Simone, who did this. Eugene Simone was the person that described many of the spiders that Perkins collected. And Simone had actually described this animal here, Dorianicus raptor. And he said it was on Kauai. And Kauai, if any of you have been to the islands, Kauai is actually quite big when you have to find a little spider. So I went all over the islands. I went to the summit, I went through the forest, to the dry forest, the wet forest. And then I was really thinking, well, this thing's really extinct. It's no more. And then I went out I had to do a survey of low elevation habitat on the north side of the island. And I thought, well, I'm not going to get much here. But then it got dark, and I'll talk about that in a second. It got dark, and everything was quiet. And then these big green things dropped down. They didn't spin webs. They don't spin webs at all. What they do is they have these long claws on the end of their legs, and they actually swipe prey directly from the air. So these, you can see kind of what, finding this totally unknown fauna was really pretty extraordinary. But the other thing about doing work in the islands and doing work on spiders, in particular the Tetragnatha, was that you pretty much have to do it at night. So this is just a film just to show you what happens. This is taken with a night vision camera. And it's very shaky because I couldn't, the night vision works great, but you can't see anything unless you shine a light on it. So I was trying to do both things at the same time. But you can see the spiders just sit there. They don't see very well. In fact, they hardly see at all. So you can just watch their behavior. You can see how they build their webs. You can see how they move around, and basically where they like to live, how they like to live. And this is what makes working on these spiders really it can tell you a lot about biology and what I'm really working on, which is kind of the interplay between ecology and evolution. So when I got to Hawaii then, it was pretty much an open playing field. There was very little known about the spiders, and so what I had to do was go into these forests where no one had gone to study spiders, and there were now reserves that had been put in place, but the reserves always wanted you to take someone, and who in the world do you take? Because they're wet, they're cold, they're not what you think of Hawaii being. So what I did then was get my mom to come along. So she was a great sport in all of this, and came with me to multiple sites. Here she is on the big island of Hawaii. And so then, unfortunately, she does live in Scotland, and so she couldn't stay forever. And so next in line came a colleague, actually, from the University of Maryland, George Roderick, and he was very good at this, and so we married each other, and that was all great. And so then we had our F1. This is our youngest kid at 1 month old. I was actually up a tree looking for spiders, and a colleague who I was with just put the net in his hand while he was lying in the forest there. But he did grow up, and he was a little bit more competent when he grew up, and he could actually carry a net. And then, you know, over time, reluctantly, he became a little bit more useful. So what I want to do then is tell you why Hawaii has been, what's so special about Hawaii for understanding diversification. I've told you about the study organisms and about the history of study in these islands, but why should people be interested in this? So over the years, the early years when I was there, Warren Wagner, a botanist at the Smithsonian, with Vicky Funk, they were both they're very good at getting people together, and they got all of the people working in Hawaii, which is on the Hawaiian fauna and flora, which is actually a very small handful at the time. But they got everyone together and put together this book in 1995, Hawaiian Biogeography, Evolution on a Hotspot Archipelago. This was really important because what they did is they put together the work of lots of different people who were really just starting out and just really trying to understand what was going on in the island. But the big thing was that it was the first attempt to test the idea that independently derived groups of Hawaiian organisms exhibit similar patterns of colonization and differentiation that relate directly to the unique geological history of this oceanic archipelago. So why is that so special? So this idea actually stems from Tusa Wilson in 1963. So Hawaii is a hotspot, and this is really the key thing about it. It's supremely isolated, but it emanates from far below the ocean, from volcanic activity far below the ocean. And what happens, and what Wilson proposed, was that the islands arose like a series of bubbles from a point beneath the island of Hawaii. So basically what happens is you have this oceanic oozing of lava, and then the islands move away from the hotspot and become more eroded and die down as they get older. So what that means is that you've got very young substrates here, you've got very old ones here. So what this has allowed then is for us to understand old communities, younger, and the very youngest starting out. And so you can see most things, at least when they've been there for the duration of the current high islands, have hopped down from the older to the younger islands. This is a general pattern. And because of this, each island represents a new start of both evolutionary change and community assembly. And this is the fundamental thing about these islands, that you can use them as replicates of different age to understand how evolution changes over time and how community structure changes over time. So just to kind of illustrate that point, marry two processes. So if we look at ecological processes, for example, immigration, extinction, things that occur over short time periods, many of the most sophisticated theories in ecology are developed for a single place, a single time. And what we want to do is to understand how ecological processes play out over time. So what we'd like to do is say, how do these ecological processes play out into the diversification of species? So really, we want to try and put these two together to understand how ecological and evolutionary processes can make the biodiversity dynamics. So these processes occur at very different, over very different timescales. This happens immediately, and this happens very slowly over hundreds of thousands to millions of years. So how do we put these processes together, and so in thereby doing, assess biodiversity dynamics? So basically what we're doing is putting these processes together to assess how biodiversity comes into being. So we look at ecological processes, in particular on the younger islands, and look at evolutionary processes going right through the chain of the archipelago. So basically what we're doing is this. You've got the hot spot here, and this is 5 million years ago. 5 million years ago, actually, Kauai just formed. The other islands were really low and far apart at that time. So when Kauai came up, it was really the only high island at that point. But then over 2.5 million years ago, you saw Oahu appear. And organisms have tended to go from Kauai to Oahu when it appeared. 1 million years ago, Maui Nui, which is Maui, Molokai, and Lanai, they popped up just next door to Oahu, and things tended to hop from Oahu down there. And so you go on with Hawaii popping out, and then this is the present day pattern. So the idea then is to look at these time slices. 5 million years ago that's how old Kauai is, that's how old the communities are. Likewise, Oahu is 2.5 to 3 million years, and then you've got younger to the youngest. So basically, you can read this chronosequence, so to speak, as a fossil record to look at the dynamics of diversification through time and the temporal dynamics of community assembly. So putting evolution and ecology together. So I want to just go quickly through then and just talk about some of the patterns that you find in spiders, just because spiders illustrate many of the kinds of patterns that you get in these islands. And I want to kind of contrast what's going on in different groups, because you think, doesn't everything just go ahead and diversify? Are there any generalities that we can make? So let's just look. How do species diversify? Looking at spiders across this chronosequence. So the first example I'm going to give is this genus Orson Welles, which was really it was Gustavo Hormiga, who's currently at George Washington University. He really liked Orson Welles, so he put it in made this new genus and described the species. That's all that Orson Welles plays. But anyway, Gustavo described these things, and then we got molecular data, which I'm not going to talk about, but we got lots of different markers. And what we did then was just look at how these things had diversified across the islands. And what you find, I'm just going to highlight here, how they've just basically hopped down the island chain. So Kauai, you get a whole bunch of species. 5 million years old, it's had a chance to have accumulate lots of species. Not quite as many on the somewhat younger Oahu. And then here, again, for this area, you get about the same number as you get on Oahu. And then just one species on the very young island of Hawaii. So basically what's happening here is you get a species that gets to a new island, goes everywhere, and then it just goes to new islands. They diversify in allopatry. They don't diversify ecologically, and you just get species accumulating. They just accumulate over time, and so you get the most on the oldest island. So this is what you call non-adaptive radiation, And the species diversify in allopatry. They're geographically separated. So colonization of the same form with no evident ecological change. So is that just what's going on? That'd be pretty boring if it was all that was going on in spiders. So let's look at some other ones. And I'll just give you a couple more examples. Here, this is work by a graduate student in my lab, Jessica Garb, and she worked on these Mecaphysa crab spiders. And so what she did was look at the ecological change through the island history. And what she found was that most of the ecology, the dark blue is Kauai, and most of the major ecological lineages had formed on this oldest island. Then what happened is they just, having formed the major ecological forms, they simply hopped down the island chain. So you get the dark blue to yellow to red, indicating they've just hopped from the older to the younger islands and down to the youngest island. So here you see diversification, ecological diversification has happened early in the radiation, and then the populations are simply hopping down the island chain. So basically what's happening here, you get lots of ecological differentiation, and then those simply go on to do the same thing on the younger islands. So you'd say here, early ecological diversification that's adaptive, and populations differentiate in allopatry. So colonization of the same form that evolved on the oldest island. And that's kind of, you say, well, that's interesting, but not that exciting. So let's just see, are there other patterns? And here's where I think it gets a little bit more exciting. You get some taxa that hop down the island chain and diversify within islands. And I'm going to go fairly quickly through these because they're pretty stunning examples, but I want to just use them as examples. So these are stick spiders, the genus Ariamnes. And you see all of these different forms. This white form that lives on white lichen, the gold-colored one that lives under leaves, and the dark brown ones that live in rocks and things. Again, we did a phylogeny that I'm not going to go into. And here you see, this is Kauai, this is Oahu, this is Maui Nui, and this is the Big Island. So they've hopped down the island chain. But what's interesting is the brown, these rectangles in brown and gold represent the different colors. So you see, I've tried to, the white one here, you can't see the white so well, but the white, the gold, and the brown have evolved repeatedly on each island. So they're monophyletic largely within each island, and they've just spouted out the similar forms, the white one, the brown one, the gold one, on each island. So repeated evolution of the same form. Now, this is my favorite group that you've heard about already, the long-jawed spiders. That one had a little bit of prey. I was actually trying to get it in the camera to focus on it. I'm not that ham-fisted. I can usually get the spider in the vial. But these spiders have diversified in Hawaii. And just to give you a feel for how they've diversified, you get a whole lineage that has abandoned web building, and these are the spiny-legged ones. And so they do what running cursorial spiders do on the mainland, and so they run around the vegetation, don't build webs. But then you get all sorts of ones that build webs and do all sorts of cool things, and they're unique to very specific habitats. So many are highly specialized, and they're altogether hugely diverse. So I've been focusing on one group, the spiny-leg clade, and the spiny-leg clade occurs in 4 discrete forms: the big brown, the green, the maroon one, and the little brown one. And again, we did a phylogeny of these things, and what we found was the green, maroon, brown, they're not each other's closest relatives. So the green, here you see the bars here represent the ecological forms. So the green popped up over and over again. The big brown popped up over and over again. The little browns popped up. The maroons popped up. But you see, they've hopped down the island chain, but repeatedly evolved the same form. So what you find here, again, is repeated evolution of the same form across species. And that is quite cool. And what we're trying to do with that is, at the moment, is try and understand exactly how that happened. So look at the mechanics of of selection on specific genes to see exactly what's going on. But rather than go into that, I'm just going to tell you one more story of diversification. And this is the Hawaiian happy-faced spider. The Hawaiian happy-faced spider that I've kind of belittled all this time because I went to study initially the happy-faced spider, but went on to this much bigger radiation. The Hawaiian happy-faced spider is a single species. So why did I say diversified? So it's a single species that occurs from Oahu down to the Big Island. And this is work I did with Jeff Oxford. And what we did was we looked at these spiders in different habitats. And what you find is that in any one population, most of the individuals are pale yellow, and the rest are lots of different colors. What we found is that there's selection maintaining this frequency of color. It's apostatic selection, frequency-dependent selection. And the idea is that birds are probably responsible. These beautiful, cute little honey creepers are actually controlling this frequency of colomorphs. So basically the idea is that when the pale yellow becomes most common in the population, birds can develop a search image for it, and then there's selection to be simply different from the most common form. We also know that these spiders don't move around, and I'm not going to go into this except to just say that these colors are based on actually allozyme data, which is sounds out of the arc, which is just protein electrophoresis data from a long time ago. But the idea that what I want to convey here is that populations at any one site are very discrete, so there's very little movement between populations. So what I did with Jeff then was look at how the color was inherited. And so we did lots of crosses, and the colors separate out beautifully in Mendelian ratios. You get the yellow is recessive to everything else, the red is dominant whatever red. And so you can cross them. The yellow allele is the most frequent in the population, so the reds tend to be heterozygous. You do a cross, and they come out half and half. And so just a beautifully straightforward system. Then we did all that work on Maui. We come over to the Big Island now, and what happens? You cross a yellow with a red front, half come out yellow, half red front, but the red fronts are all male, the yellows are all female. And then you can actually do inter-island crosses. If you do a male from Maui with a female from Hawaii, they're both yellow, and the males come out like the Hawaii males, red front, and the females yellow. So what has gone on here? It seems that they've colonized Maui, and we know that based on molecular data, and then they've gone on to colonize the Big Island. And it seems that probably what happened is just a few individuals would have been involved in the colonization. And then there would have been selection to recover the diversity of the initial population. So selection to recover this diversity by a different genetic mechanism. And again, this is something that we're currently looking at, and I'm not going to go into any detail here. So what I want to just highlight from this is just to say that on each island you get different patterns of diversification. So we looked at the Orson Welles, where you got colonization of the same form with no evident ecological change. The Mecaphysa, the crab spiders, colonization of the same form that evolved on the oldest island. Tetragnatha, repeated evolution of the same form across species. And then the happy-faced spider, repeated evolution of the same form within a species. So what I want to do just in the last 10 minutes here is then go on to say, if we realize now that species can form differently, so then how can you use the islands and the chronosequence to look at how communities assemble? So again, let's look at this wonderful sequence of islands, 5 million down to 0 years old. This sequence has been studied from an ecological perspective for quite some time now by Peter Vitousek. Peter has been looking at what he called the LSAG sites, the long substrate age gradient. and so has looked at sites from Kauai down to the Big Island to see how soil stoichiochemistry changes. And what he's found is that you get really clear patterns that on very young substrates you get very low nitrogen, and then on older you get very quick accumulation of nitrogen, and then they die down. And the important thing here is just that it shows you that the stoichiochemistry has a sequence. It changes over time. Now, we wanted to see, first of all, whether this sequence was reflected in up the trophic levels. So I have a student, Susan Kennedy, who was interested in just seeing how this pattern might be reflected in the plants and the arthropods. So what she did was she looked at the spiders and the plants, and here what you see is the very youngest site, and here are plants, and then different groups of spiders. These are the Ariamne spiders in the light blue, the ones they actually feed on other spiders, so the highest trophic levels. So you can see this increasing, in one site, you get an increase in the nitrogen content based on isotope ratios that reflects the higher trophic level. Now, what's really cool is that as you go up in age, you get just an increase in overall nitrogen, reflecting very precisely what Peter had found in terms of the soil stoichiochemistry. Now, that's not hugely unexpected. The site has higher nitrogen, so why should you be so surprised? I think the most interesting thing is if you look at these two sites here, you see the big differences in in the nitrogen content of the spiders. And these sites are it's right here on the Big Island. Now let's just zoom in. Can you really distinguish them? You can distinguish them, but they're right beside each other. And so you get a signature very clearly of the substrate age reflected through the trophic levels of the spiders, even though the spiders are living right beside each other. That really highlights the kind of how presumptive many of these organisms are. So what we've been doing then is looking at overall across the chronosequence, how are there general patterns of species diversity as you go from older to younger islands? And here is what's quite cool. This is work that I did with Bruce Baldwin, and we looked first at plants and looked at the number of species per unit area as island age increases. And for plants, what you find is that bigger lineages tend to go very high and then level off. Smaller lineages tend to keep plugging along. And you find that with animals as well. Different groups of animals that we looked at, bigger lineages go very high on the young islands and then drop down, and the younger lineages keep plugging along. This, interestingly enough, is a pattern that we find in many groups of spiders. And rather than highlight this, I want to just summarize the different groups of spiders and which ones show different patterns of increase. Here you have our friend Orson Welles, the highest species number on the oldest island. Remember, this is non-adaptive, and they just keep plugging along, and the highest number of species on the oldest island. Mecaphysa reach a very high number fairly early on. And then in Tetragnatha, and Ariamnes, where they repeatedly evolve the same form, they come back together in sympatry almost as soon as they've formed new species. For these, you get the highest number of species early on, and then they drop down on the older islands. So basically what you're seeing here is that some groups that in the spiders that don't undergo adaptive radiation, they just keep accumulating. The Mecaphysa accumulates quickly and then levels off, and then these ones that diversify truly repeatedly show this peak of diversity on the younger islands and then drop down. So let's then look, given that it's the youngest island where you start to see things really starting off, let's just look quickly at the youngest island. And this is where we've started our current work that I'm just going to mention here. This is Hawaii Island, the youngest island. And Ham Carson even said, we suggest that the youngest island at any one time has always been Hawaii's major evolutionary crucible. So we're trying to understand exactly what's going on this island at the moment. Just seeing just the role of this is a geological map. The substrates vary from the very young substrates here. This is a continuously oozing volcano of Kilauea, and you get onto the oldest substrates up here. And so you get them continually fragmenting and coming together. And what we're doing currently then is trying to see whether you get signatures of establishment of communities on these very young substrates, and whether they change predictably as they get older. So what we're doing at the moment, and this is, as Diana said, we've got a lot of people in the lab that are currently involved in this effort. What we're doing is we're trying to standardize for elevation, precipitation, the kind of forest, and also making sure that they have reserves that are available to us. And what we're doing then is looking at these sites, and they overlap a lot with the sites that Peter Vitusek used to look at the soil stoichiochemistry, and so it allows us to make maximal use of the information available. So we across these sites of different age, what we do is we collect arthropods, and we collect a lot of arthropods, and in a lot of different ways. So we use beating samples. This is a graduate student there who's they do time beating and specific vegetation types across the different sites. This is a malaise trap going up into the canopy. This is one of the students climbing up a tree where they're doing canopy clipping. So we get lots of gazillions, to be honest, of different of samples of arthropods. And what we're trying to do is apply macroecological theory that I'll just very briefly touch on here, but this is the work of Andy Rominger, a graduate student in the lab, who's shared with this guy, John Hart. And basically what they're trying to do is look at the predictability of species abundance distributions. The theory kind of, all it does is look at how energy in a system can be most parsimoniously, in terms of statistical kind of packaging, how it can be most parsimoniously packaged into individuals and then species. And so you develop predictions based on this theory, and then you look at deviations from that theory, and that allows you to tell where something might be going on. If you get a deviation from statistical expectations when there's no biology really going on at all. So what they've done now is look at certain sites. Again, this is based on minimal data at this point, but what they found is looking across sites of different ages, you can see the oldest sites. This is for detritivorous arthropods, and what the black line is the prediction, and the little circles are the actual data of arthropods, detritivorous arthropods at that site. And you see the younger sites give the higher deviation from these predictions. This is based on very preliminary data, just to give you an idea of what we're trying to do, but it's one of the main things that we're trying to figure out at the moment. So what we're also trying to do is look at predictability in terms of food web interactions. So see how species interact on the very youngest substrates when they're just colonizing, and see whether there's a predictable pattern in terms of, do they become more specialized? Do you tend to get more predators initially or later on? How are the predictable components of the assembly and the interactions? So what we're trying to do then is use this dynamic geomorphology of the youngest island to determine the importance of the changing functional roles of taxa within arthropod communities as they differentiate and as they assemble. And then feedbacks to and from that dynamic community are presumably fostering diversification, which we're measuring at the same time. So essentially, we're trying to synthesize ecology with evolutionary biology to understand differentiation and diversification in long-term community assembly. So this is what we're doing. And in the last few seconds here, I just it would be a mistake to just leave you with an idea of this is just the most outstanding island to do all of these things. It's so isolated and pristine and amazing, which it is, but it's also now one of the most visited destinations of any. So it's very isolated up in the higher elevations, but at the moment it's really basically become, the Pacific Ocean effectively has shrunk. So the native communities of Hawaiian organisms emerged when evolution outpaced immigration. Alien propagules now arrive faster than ever. So the thing that we're trying to get at is how are communities responding to the new dynamic. So just to give you a feel for invasive species, I'm not going to go into these except to say there's lots of them. This is ginger, an invasive plant. This is the Nature Conservancy habitat. What you don't see is a fence here. Now the fence is not keeping the ginger out, it's the people that are working their I won't say what off, to try and keep the ginger out. It's like this army trying to get into this site. So basically, we've got a huge problem there, and what we need to do is understand the dynamic. What happens when you've got a community that's put together through evolutionary processes, and then you stick something like this into the mix? These continually arriving new propagules. So sometimes you think, well, this is just hopeless, and you feel like just giving up and saying to heck with it. You know, as one worker in Hawaii said to me recently, we need people like you, Rosie, because someone has to describe these things before they go. And that's kind of depressing, to be honest. And so I just think of the Lorax. Catch calls the Onceler. And he lets something fall, it's the last tropula seed, the last one of them all. It's very sad, but the bottom line is there is hope out there, and you sometimes you think, well, you know, is there really? But actually this is a colleague, Art Medeiros, and what he did was, in the south side of Maui, just completely devastated, and this is, when I arrived, it was just this pasture with a few trees. All of these trees are native, they're endemic, they're unique to that site. What Art did single-handedly was put a fence around this area. We figured out it was kind of the maximum area he could handle, and then physically pull up the weeds and plant inside the fenced area. And this is it now. I mean, it's over time. If you look on Google Earth, you can see these big, very regular patches, and this is where he's doing this effort. And so things are coming back, which is really pretty remarkable. So with that ray of hope, I hope I've convinced you that the Hawaiian Islands are the most extraordinary places, extraordinary place to work. That all islands are really pretty outstanding places to work, but the Hawaiian Islands in particular, because of the chronological order, allows you to integrate evolutionary and ecological processes and hence understand biodiversity as it's the dynamics of biodiversity as it's happening. And these are the people that are currently involved in the project that we're working on, and there's a bunch of them there, but they're all tremendous colleagues. But I just wanted to thank you and ask if you had any questions. If I've got any time left, I'm sorry I've gone on for a bit. So thank you.